Hi guys, this is Rohan C. from Bio 418. I'm doing an experiment involving capillary action, and this is the setup for the experiment. So I have a white flower. White is important because if you can't see the color coming out from the capillary action. This is blue colored water. It's made by this dye from this packet, McCormick assorted color. I think it's kind of old. I started up with this fork, a bit dirty, but I'm using it for all of them. So I recorded what date and time I placed it in, and basically that is basically the setup. Now this is going to be, there will be 5 plants placed at room temperature and 5 plants placed at freezer temperature. So what this experiment is going to try to show if the temperature affects capillary action on the same type of flower, the same type of dye and color, which is very important. And so yeah, a bit more detail about this experiment. Well, this experiment, for the water at least, there was 3 drops of dye, blue dye, and for this little contraption, it's 2 paper clips like these, unbent completely and taped over on the sides, and I put the stem right through it and just dipped it down. I'm not going to take it out now, obviously that would be a confounding variable. I cut all the plants with this scissors, this pair of scissors only, and hey, so this is back to the lab of capillary action at different temperatures. This is my whole setup. So there's 10 plants here on the table. It's room temperature. And actually this part of the house does not get air conditioning, so it's been the same temperature the whole time the test is run. Here we have our 3 mist trials. What happened is that apparently in the fridge you can freeze water if it's there for about 4 days. So you can see as I'm touching it, there's ice moving in there. Now that caused the plant to die, and honestly there's no blue there anyway. So I'm going to use day 1 trial data only. Now this same thing here, only half of it died, so it's about to die really fast. Sorry about that, yeah. And this one was a mistrial as we were moving this to a different shelf in the fridge. I'm not sure you can see it, but there's a bunch of blue dye. That's because it was splashed by the water. Now, this is our fridge. Now, in the fridge, we have 10 plants, well, now 7, because of the 3 mistrials. And as you can see, probably, that there's a lot less blue dye on the petals. So from this initial observance, you can tell what part, if my hypothesis was wrong or not, if capillary action is affected by temperature. Now, in the same region about here, so it should not be that. I just moved them around because I was taking data. I'll probably be dumping them out now because it's been 2 days. Right, so back to the table. As you can see, there's 10 here, and even the camera can see that that is a very blue petal. It's very concentrated. You can see that that petal needed a lot of water and it got it, and it's now blue dye. It's a lot more than the ones in the fridge. Here's one here, and here's another one. Oh, and this is a very good example because there's so much on the base petals than the middle. It really shows you how the distribution of water is during capillary action. Now this is my data sheet. There's plant number, what's it called, percent. This was I judged, so I judged how much percent of the plant is blue, and these are the days. Those are observances, or like explanations for mishaps. As you can see, there's one here. Actually, plant 13 is interesting. It had frozen just like the mistrials, but it's very much alive. It is a lot healthier looking than some of the other plants that did not freeze, which is pretty weird. Now, this is a mistrial. That's the one that was splashed back onto. That was before we even could take data from it. And that one is one of the frozen ones. And that's it. Yep. So that is pretty conclusive. So over the course of this experiment, I was, thinking about how exactly does the blue dye travel up the flower, right? So I did this is not from the actual experiment. This is just, there. But, so I let this sit one day. I saw some really good results from one day's. I don't see much blue on the stem, the petals, but I guess that's okay. So basically right now I'm just dissecting this. I'm just gonna cut this right now, the stem from the petals. There we go. This is the flower. Now, the camera cannot see this because of the really bad lighting, but here there is discoloration on the skin of the stem. So what I'm going to do now is I'm going to open up the stem. So again, I really doubt the camera can see this, but there is blue in the middle of the stem here. Like, very fine, but you can see that there is blue. It's not green, it's not the stuff that we've been finding. And I'm removing the skin to get rid of the light, And so, yeah, there is blue. Well, it may not be blue, but it is definitely not green. It's not supposed to be there. So anyway, I conducted this experiment just because I wanted to, and might as well, right? And so I was noticing that at the base of the petals there was a lot of blue. So that's why, it's just, at first when I was looking at the experimental results and looking at the petals there, I saw that this tip here, see, this is the petal. The tip here, I was looking into the flower without it actually taking it out. I saw, it was darker than usual. I thought maybe it's just the lighting. But then one day a petal fell out as I was taking it out, dumping them out, and I just, wow, it's blue, right? So